afternoon, sir.
Okay.
This call is now being recorded.
So, have you gone through the material I posted in module with respect to potassium channels?
One video, sir.
One video. Okay. So, we will try to go through it. Try to complete it a bit fast because I want to complete a bit portions quickly. So that's why if you go along with me, then only I can push the classes a bit fast. Otherwise, we'll lag behind. Because maybe after 1 or 2 months, you will come back and directly we will go for exams, practicals, etc. We may not get time for theory and all. So that's why I will try to finish up the theory as early as possible. Okay. So I'll be presenting. Today I'll be taking mainly potassium channels, and if time permits, I will discuss a bit about chloride channels and calcium channels. Okay. Uh, okay. Coming to potassium channels, unlike voltage gated sodium channels, voltage gated potassium channels, at least there are four different types. Depending upon the type of literature you go through, either it some says it is three different types of potassium channels, other some says four. So I have taken in the class also I have taken four different the classification where there is four different types of potassium channels. So that's what we will discuss today. And I'm now discussing only the basic structure of a potassium channel. It's not that every potassium channel, I told you there are about three or four different kinds of potassium channels, depending upon the source you refer. Whatever maybe it's either three or four. These different kinds of potassium channels differ in their structure. It's not uniform. Sodium channels, it's uniform. But potassium channels, we have different types of potassium channels, that means different types of channels which allow the movement of potassium ions. And the structure of each channel is, each type of potassium channel is different. This is one just a typical kind of potassium channel I am discussing. Just like sodium channels. How many domains are there in a sodium channel? Do you remember? How many domains?
Yes, all the
Four domains. Yes. Similarly, here also potassium channels are also having four domains. That's why it is called as tetramer. But unlike sodium channels, each domain is having only two transmembrane helix. What is the number of transmembrane helices in sodium channel? I can't. You can type it, or if you. What is the number of transmembrane helices in a sodium channel? Or you can unmute. I am six. Yes, Bindu is correct. It's six. Yes, we have six transmembrane helices, but here we have only two transmembrane helices. That is one typical case, but there are certain potassium channels which have the structure exactly similar to sodium channels, where we have six transmembrane helices. But I am discussing about mainly about one kind of channel wherein we have only two transmembrane helices, and each domain will form a pore forming domain. That means one transmembrane helix from each domain forms the pore or surrounds the pore, and these four transmembrane helixes from these four domains has the selectivity filter. And this selective filter, what is the selectivity filter in sodium? What is the selectivity filter in sodium? The selective filter contain what amino acids? Deca, yes, good. Here it is T, V, G, Y, G. D is threonine, V is valine, G is glycine, Y is tyrosine. So this selectivity filter contains threonine, valine, glycine, and tyrosine. Again, it is negatively charged. Then only it can interact with the positively charged potassium ion. So I'm repeating, similar to sodium channel, which has is a tetramer, that means four homologous domains. Each domain we have two transmembrane helices, and one out of these two, one transmembrane helices forms the pore, so that the four domains will have one transmembrane helix each forming the pore, surrounding the pore, and we have five amino acids: threonine, valine, glycine, tyrosine, and again glycine, which are negatively charged. That forms the selectivity filter, and that selectivity filter is the one which permits only potassium ions to move, move through the potassium ions. Then let's see how it occurs. See, this is a single domain wherein we have two transmembrane helices and we have the loop, and this loop is having the uh, uh, amino acid sequence TBGYG. Okay. See, here you can see what I told. Here you can see one, two, three, and four domains, and this transmembrane helices, this one, and this one. It forms the four. Here it is more clear. You can see the two transmembrane, one, two, one, two. This is one domain. This is second domain. Another domain will be in the front. Another domain will be in the back. So here we can see left side, right side, back side we can't see, front side we can't see. But each domain will be having two helices, and the loop connecting these two helices, we call it as the T loop, wherein we have the amino acid sequence which act as the ion filter. Then I told yesterday, last class I told that in the sodium ions there is Becker filter which permits only sodium ions, which doesn't permit calcium ions. And potassium ions, one thing is that potassium ions is slightly bigger in size, so it can't also move through it. But since the opening is slightly bigger, sodium ions can easily pass through. Then how sodium ions are selectively filtered out? Only potassium ions are selectively allowed inside. How does this happen? This is another.
Ion selectivity filter having different amino acids.
No,
So their deca is responsible for forming ion selectivity filter, but here is different from sodium, right?
actually no. There also it is. Yes, amino acids are different, but the selection occurs due to another reason, not due to the difference in the kind of amino acid. No. I will, yes, it is
Calcium.
because of the water molecule, because of the difference in the hydration. Again, size does matters, and the water of hydration. I will show you the videos of it. Here is again another picture wherein we have the transmembrane helices. We have the selectivity. See, the potassium ions. So either it is potassium or sodium. It is it is an ionic in nature in the solution in the hemolymph. So these ions will interact with water to form a water of hydration. That means it will be surrounded by water molecules. I will show that picture. See it over here. Just a minute. See, this is potassium ion, this is sodium ion, positively charged. What you see, this B, this is all these things are water. Two hydrogen over here, this is oxygen. You can, you know that oxygen is negatively charged. So, these four water molecules interact with potassium. So that means potassium ions as well as sodium ions will invariably be surrounded by water molecules in the hemolymph, that is in the solution. But once it has to pass through this selectivity filter, it has to remove the water of hydration. That you can see. See, this is this violet colored thing is the ion. What you see, that blue color which is surrounding just like a halo around this violet color, that is the water of hydration. So once it is passing through this selectivity filter, it has to remove the water of hydration, then only it can pass. So it removes the water of hydration, it passes through the selectivity filter. Once it reaches out the into the cell, crossing the selectivity filter, it again becomes hydrated. So what happens is that once it is passing through the selectivity filter, it has to remove the water of hydration. And during this passage of this ion through the selectivity filter, it will interact with the negatively charged amino acids in the selectivity filter, TBGYG. So it will interact with oxygen ions, negatively charged oxygen ions in the selectivity filter. But see here what happens. Potassium, because of its larger size, it can effectively interact with all the four negatively charged oxygen ions in the selectivity filter TBGYG. But take the case of sodium. The size is small. It can interact only at a time. It can interact only with two oxygenatively charged oxygen. It can't interact fully with other oxygens. So this is how the sodium is unable to pass through the selectivity. Whereas potassium, which can effectively interact with all the four transmembrane helices in the selectivity filter, it can pass through, while because of the smaller size, sodium cannot pass through. Clear? Yes. If there is a need to repeat, I will repeat.
Sir, വാട്ടർ മോളിക്യൂളും അമിനോ ആസിഡും തമ്മിലാണോ ഇന്ററാക്ട് ചെയ്യുന്നത് അതോ
No, 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 no. There won't be any water molecule once the ion is passing through the selectivity filter. In the selectivity filter, we have ions, we have amino acids, and amino acids we have oxygen ion, oxygen atoms, which is negatively charged. So these ions, once it removes the water of hydration and enters the selectivity filter, these positively charged ions interact with the negatively charged oxygen atoms of the amino acid in the selectivity filter. Bindu, any doubt? Okay. So that's how the selectivity happens for this channels, how it allows potassium to pass through, how it doesn't allow sodium to pass through. And I told you there are about 4 different kinds of potassium channels, and let we discuss what are the different kinds of potassium channels. One kind is voltage gated potassium channels. The structure is almost similar to voltage gated sodium channels. Then there is inwardly rectifying potassium channels. We discuss about inwardly rectifying potassium channels. The structure of it is similar to what we had discussed just now. 4 domains with 2 transmembrane helices. Then we have the next one, tandem 4 domain potassium channels. This is the one which is least understood. So that's why certain literature avoids this as a type of potassium channel. So when they say we have only 3 different types of potassium channels, certain literature says that there won't be tandem 4 domain potassium channels. They will describe only voltage gated, inwardly rectifying, and ligand gated potassium channels. Ligand gated potassium channels, it mainly responds to calcium ions. We'll see it one by one. How does it differ from one another? Then the first one, that is the voltage gated potassium channels. The structure is entirely similar to what we have studied in the case of sodium channels. We have four domains. Each domain will have six transmembrane helices, and this among the six transmembrane helices, the S1 to S4, especially the S4 transmembrane helix, is the voltage sensor, and S5 and S6 are the forming domains. I repeat, we have four domains, six transmembrane segment helices in the each domain, of which S4 especially is the voltage sensor, and S5 and S6 are the co-forming domains. The same thing structure we have studied in the case of sodium channel. Then comes inwardly rectifying potassium channels. See, when we discussed about this channel, what is it? If you remember the transmission of ions, when there is action potential, sodium channels opens and sodium channels move from where to where? Outside the cell to inside the cell, or inside the cell to outside the cell?
Outside.
Outside to
Outside to
inside.
inside.
In potassium channels open, and then potassium will move from
Inside to outside.
inside to outside. But here, these channels permit the movement of potassium ions towards inside the cell. That's why it is called as inwardly rectifying potassium channels, because these are the ion channels which permit the movement of ion channels into the cell, not outside the cell. Till now, what we have discussed is which permits the movement outside the cell, but this is the one which permits the movement into the cell. That's why it is called as inwardly rectifying potassium channels. So, this inward movement of potassium ions happens during hyperpolarization. I, just for your recollection of information what you have studied, I just put out a picture. So this is the rising phase of action potential that you might have studied. This is called as the depolarization phase, wherein the sodium channels open, all the sodium ions move in. Then once it reaches about minus plus 35 millivolts, then the sodium channels close. But that we have seen how the sodium channels inactivation gate of sodium channels closes it. Sodium channels responds very slowly. It can be now you can consider two persons. Whenever there is an activity, there are certain students which come forward quickly, while there are some other students which comes forward pretty slowly. So sodium ions opens and closes pretty fast, while the potassium channels open slowly and closes slowly. So once the depolarization reaches about 35, then only sodium ion starts responding. Sodium channel starts responding. Sodium channels open. Potassium ions start moving out, and this is the falling phase of action potential, and it is the repolarization phase. That means again the membrane will get repolarized. But this is the resting stage. Actually, it should be minus 70. It should be not. It's not minus 90. It's minus 70. So once it reaches the resting stage, okay, it's fine, it should have been stopped here. But what happens is that at this stage, again there is a downward, again there is a repolation. What will happen is that at this time, the inwardly rectifying potassium channels open, and more potassium ions move inside. That is because of the opening of this inwardly rectifying potassium channels. See, you can see it over here written that voltage gated potassium channels remain open after the potassium potential reaches the rectifying resting level. It's not that the common potassium channels open. It's the specific inwardly rectifying potassium channels that remain open. So the inside, the concentration of potassium ions slightly increases than when it is in the resting stage, because of the higher concentration of potassium ions permitted by the inwardly rectifying potassium channels. Fine. Then how come this inwardly rectifying potassium channels remain closed till this stage, till the repolarization reaches the resting potential? During the depolarization stage and the initial phase of repolarization stage, what will happen is that uh, this channels, this inwardly rectifying potassium channels, are blocked by magnesium ions and polyamines. I don't know the exact way by which it blocks the ion channel and how that block is opened during the hyperpolarization phase. I don't know exactly how it happens, but it is described in literature that during the depolarization phase and the initial phase of repolarization, this channels, this inwardly rectifying potassium channels are blocked by magnesium ions and certain polyamines. There are certain literature which says one specific polyamine. I forgot the name of that polyamine. I think in the literature which I have given you in the Moodle specifically says about that, but name of that particular polyamine I forgot. Okay. And the structure of the inwardly rectifying potassium channels is the what which we described as discussed as the general structure of that potassium channels, wherein we have four domains, each domain having two transmembrane helices. That what we discussed, that is the same structure of the inwardly rectifying potassium channels. I will show it. Okay. This is voltage gated potassium channels. You can see the same structure as that of sodium channels. Six transmembrane helices, fifth and sixth forms the four is the voltage sensing one. See the inwardly rectifying one. We have only two transmembrane helices. That's all. And this loop is called as the T loop. And here we, if we see from the top, you can see four domains, and in the between the four domains we have the S5 and S6, which forms the four. And this is with respect to the inwardly rectified. One thing you can see that everywhere, if you see the literature, with these different types of potassium channels is given a particular kind of abbreviation. This voltage gated potassium channels, it is always represented as KB, and that inwardly rectifying potassium channels is KIR, inwardly rectified. Usually it is KIR and all that told usually there. So inwardly rectified is KVIR, this is KB, voltage gated. So this is with respect to two voltage gated and inwardly rectifying potassium channels. And the next one is tandem four domain potassium channels. Tandem four domain potassium channels. This is one of the least understood potassium channels in biology. Its structure is not that well understood. Its function is not that well understood. That's why the opening here in the first sentence it is written that it has variety of mediators. That means its opening and closing is controlled by several things. It can be ions, pH, lipids, regulatory proteins, etc. It's not that voltage gated. It's not that ligand gated. So people has not yet clearly understood what controls the opening on and closing of potassium channels. It's also called as leak channels. We are, if you see literature, ion channels, we discuss mainly about gated channels, where there is a control of the movement of ions. In some basic classification of ion channels, classify it as leak channels and gated channels. Leak channels and gated channels. Whatever we are discussing is gated channels. That means there is something to control the flow of ions, isn't it? It can be voltage gated, it can be ligand gated, stress gated, whatever it may. Leak channels is nothing but it's an open gate. Just like most of the time our gate of Kerala Agriculture University, most of the time it is a leak channel. It remains open almost all the time. Rarely it becomes gated. The security person closes the gate. Compare the entrance of a boys hostel and a girls hostel. Voice hostel can be considered as a leak channel because it is open every time. Anyone can enter and move. But ladies channel is gated. A security guard will be there which controls who is going out and who is moving in. So leak channels doesn't have any control. So this is considered as a leak channels, and it almost always allows the movement of potassium ions into and outside it. So that's why it is called as leak channels. And again, it has four transmembrane segments. I will show the picture. Yes. This is the structure of a tandem pore domain potassium channel. Say 1, 2, 3, and 4. We have, that means we have four transmembrane helices. And you can see the loop and the opening between 1 and 2 and 3 and 4. So that means it forms two openings, two pores. So that's why it is called as the tandem pore domain. It will have two pores between 1 and 2 and 3 and 4. So that's why it is called as a tandem pore domain potassium channel. So we have four transmembrane segments with two ellipses and has two openings. So that is with respect to tandem pore domain potassium channels. This is a picture which has the structure of all the three. The first one, which is the first one, with two transmembrane helices, inwardly rectifying, and the second one, which is four transmembrane helices with two openings, it is tandem pore domain. See here again the abbreviation K2P. That is because we have two pores, two openings. Tandem pore domain potassium channels. Then this is the voltage gated potassium channels KV. Okay. Then comes the next one, ligand gated potassium channels. This ligand gated potassium channels, it can vary. It can have either 2 or 6 transmembrane segments. It works with calcium channels. That means calcium ions controls the movement of opening and closing of these potassium ligand gated potassium channels. That's why it is called as ligand gated potassium channels, because calcium controls the opening and closing. So this is all the four together. This is voltage gated. This is calcium activated. It's not that perfect case. Here we have 6 transmembrane segments, not just like sodium voltage gated ones, but the structure is slightly different. This is inward rectifying potassium channels. This is tandem pore domain potassium channels. Okay. Now I will share. I will just show the videos a bit quickly. I will explain that also.
Voltage gated potassium
You can see
channels.
this.
In a previous video, I talked about the activation cycle of voltage gated sodium channels, how they're able to proceed from the closed to open to the inactivated state back to the closed state, and talked about the various factors that are involved in those transitions. For this video, I'm going to talk about voltage gated potassium channels, keeping in mind that there's a vast array of potassium channels belonging to the voltage.
See what he is telling. There is a vast array of potassium channels. That means there are different kinds of potassium channels.
Gated potassium channel family. The one I'm going to focus on are the potassium channels that are classically associated with the neuronal action potential. And probably the easiest way to discuss these is to talk about them in relation to the structure and function of the voltage-gated sodium channels I covered earlier. So if you're not familiar with voltage-gated sodium channels, or you haven't seen the video that I put together before, might be a good idea to follow the link and check that out first.
This is why I took sodium channel first, because sodium. Once you understand sodium channel, then understanding other ion channels is pretty easy. That's why almost always structure of sodium channels is the one that's pretty well understood and it doesn't have much variation. If you start with potassium channels, it has 3 or 4 different kinds, then it gets complicated. That's why we always start, whenever we discuss about ion channels, almost always all the lectures, if you see in YouTube or anywhere, starts with sodium channel.
So voltage gated potassium channels share a number of structural similarities as to the voltage gated sodium channel counterparts I discussed earlier. Just to go over these quickly, voltage gated potassium channels are comprised of 4 subunits, where each subunit has a pore forming domain and the voltage sensor domain. So for the pore forming domain, a couple of important features is it has the selectivity filter, which helps
What they had discussing is the voltage gated potassium channels, wherein we have 4 domains, and in each domain we have 6 transmembrane segments, and in the all the 6 S4 is the voltage sensing transmembrane segment. That's what you see it over here. This is the voltage sensing one. That's why it is pushed downwards.
impart a high degree of selectivity for potassium ion. It's also in the pore forming domain where the voltage gate is located. And just like in the voltage gated sodium channels, the opening and the closing of the voltage gate is coupled to the position of the voltage sensor out here in the
Yes,
voltage
that's
sensor
what I
domain.
told.
One
Voltage
important
sensor.
structural and functional difference that these potassium channels have relative to the sodium channels is they lack in an activation gate.
So there is
Because
no
they lack this
inactivation gate.
inactivation gate, their activation cycle is a little simpler. First of all, the voltage gated potassium channels exist in either the closed state or the open state. The potassium channels are in the closed state when the membrane potential is relatively negative. This pulls those voltage sensors, the positively charged voltage sensors out in the voltage sensor domain, inward towards the inner leaf of the lipid bilayer. This causes the voltage gate to be in the closed position, which includes movement of potassium down its electrochemical gradient, typically from inside the cell to outside the cell. When the membrane potential depolarizes, this allows the voltage sensors to relax upward, leading to the opening of the voltage gate, which allows for potassium efflux. For the channels to return to the closed state, this requires membrane repolarization. This membrane repolarization is often accomplished by the activation of these voltage-gated potassium channels. In other words, when these potassium channels open, leading to that outward potassium movement, that's typically what causes the membrane repolarization as the positive charge is leaving the cell, making it inside negative. This causes the voltage sensors to be pulled back inward towards the inner leaf of the membrane, and the voltage gate slams shut. One important feature for this activation cycle is that the transition between the closed and open state and back to the closed state for these voltage gated potassium channels is relatively slow. Another name for these channels
That's
are
what I
delayed
told. The sodium channels are pretty fast. Potassium channels respond very slowly.
rectifier potassium channels. So this is in contrast to the voltage-gated sodium channels. When a membrane depolarization occurs that leads to channel opening of the voltage-gated sodium channels, the voltage sensor moves outwards and the voltage gate opens
It's
relatively
fast. Sodium
quickly.
channels are
In
pretty fast.
contrast, for these voltage-gated potassium channels, when membrane depolarization occurs, it takes a few milliseconds or so for the potassium channels to ultimately respond and achieve their highest level of open probability given the magnitude of the membrane depolarization. And just like they are slow to open, they also tend to be slow to close. When the membrane potential is repolarized, you tend to see a persistent potassium current from these voltage gated potassium channels. This persistent potassium current can lead to a prolonged membrane hyperpolarization, which is often observed during the latter stages of the action potential.
Okay. This is basic thing about the channels. Then next I will show you how the selectivity filter works. I think it is
function relationships. It's such a huge, important topic and idea that runs through everything that we study in biology, from, just almost anything that you talk about, the structure of mitochondria and how that helps with the function that it's supposed to carry out, the structure of my small maximizing absorption example, under idea, and it's something you should. Nerve cells or neurons transmit information. They transmit a signal, a sensation, or a pain to help you actually do something about it. So if you zoom in to the axon of one of these nerve cells, this is
this
what
is a basic thing what they are discussing. I don't know whether it's visible. It is potassium channel, sodium channel, sodium potassium ion inside, sodium ions outside. So when it is depolarized, sodium ions move in. So you can see the violet ions all inside. Then potassium channels open, all potassium ions move out.
comes out sodium.
One thing, the difference in this video is they're telling about an inactivation gate over here, just like the inactivation gate of sodium channels, but none of the literature says about an inactivation gate. But so I doubt whether there is an inactivation gate in potassium channels as in sodium channels. But this video tells about it. I'm not sure
And we're
about
going to
it.
look at the structure of the potassium channel, which is kind of what we're looking at over here. It's kind of like a ball and tunnel type thing. So you can see when this ball is attached in this particular place, it has the ability to exist in a couple of states where the channel is actually closed, or where this ball can literally block the channel and the channel can be not activated. So it turns out when no messages are being transmitted across a nerve cell, you end up with no movement of ions. But when a message is being transmitted, you have sodium that rushes in through sodium channels, and then you have potassium that rushes out through potassium channels. It flows out down its concentration gradient through a process called facilitated diffusion, which you should understand if you talked about any kind of membrane transport already. And the process of potassium rushing out is called repolarization. So once again, it's going to be hard to understand the significance of all of this unless you fully understand how an action potential actually works. But don't get bogged down by this. Right now, we're just focusing on the structure of this particular potassium channel and how it actually helps with its function. What is its function? Its function is to allow potassium to actually move out. So here you can see, actually, this is not the correct diagram. This diagram is showing a supposedly end up with, you have potassium ions that will basically diffusion, but as we're using a, so that's, it can be closed and it can be blocked by this globular subunit. So if this globular subunit, a glob is just a fancy way to say a ball of amino acids or a protein or a polypeptide, can actually physically block the channel, and therefore it's closed. When we don't need it, when we don't need to send a message, it can actually be closed off. When we need it, it can open up and the ions can flow by facilitated diffusion. So structure and function of potassium channels in axons. Axons are the long part of a nerve cell. So if you have a nerve cell, oh my gosh, this is not going to look pretty. Not going to look pretty. But there's a nerve cell. This of the is the axon, and this over here.
I will show the next. I will show the next one. Next one will clearly shows how the selective filter works.
over the potassium channel. So here's the channel right here. And we will have some sodium ions, which are those, or potassium ions, which are those, and sodium, which are these little green guys. And it would seem like this green guy could easily fit right through this channel. But that's not the case. You gotta remember that these are charged particles, so they are positively charged. And what are they floating around in? Well, they're actually floating around in water, so we need to add water to these. So we'll add some water. Now we have water added to them.
This is what we seen in that slide. The positively charged sodium and potassium ions will be surrounded by 4 water molecules.
And we're going to say that you need 4 oxygen atoms for every ion. Doing it just to show how it works. So now, this sodium, it needs to have 4 oxygens attached to it, and it can't easily fit through here. What happens is, water actually can't go through this tube.
See what they're saying, water cannot move through it, so it has to remove the water of hydration. Whatever be the ion, sodium or potassium, if it has to pass through the ion channel, it has to remove the water.
Water cannot go through. Can't go through. And the reason is because of these oxygen atoms right here.
See,
They
because of the oxygen atoms, I told you there will be oxygen atoms provided by the amino acids in the selectivity.
prevent water from going through. So if water can't even go through, what can? Well, it's actually these sodium ions, or these potassium ions can. So if we had a potassium right here that just randomly bumped into it, and it had some water on the outside, right there, so we'll add some water molecules. It has 4 oxygens attached, or interacting with it, that will balance its charge. And that's good. But could we have, say, maybe a sodium ion do this? Let's draw up a sodium.
See in this video, see the size of potassium and see the size of sodium. This larger ones are potassium. The smaller ones are sodium.
Ion. So sodium ion. And it cannot interact with both of these. It's too small. It can't interact with that one and that one at the same time. So it can never really get the 4 oxygens that it needs to balance its positive charge. So that's why sodium can't go through this channel. Only a large ion like potassium can. So we had our potassium ion going through. It started right here. Now it's going to squeeze through and go right here, and it's going to lose all its water. And at this point, it's just going to keep on going through. So it's going to be right there. And when it's past this first step, another one can come behind it and start interacting with it. So draw the red. And again, the water would just pop off as this one goes down, and as that one goes down again. So now, what it would look like is this, where this potassium ion is interacting with this oxygen, this one, and this one, where this one's interacting with this oxygen atom, this one. So our potassium ion still has 4 oxygens interacting with it. Now the reason it goes down is just because of concentration gradients. It just randomly bumps into this. So let's say you had some potassium ions floating around, and they have the water around it, but I'm not going to draw the water anymore. Let's say there's also some in here. It comes down to how often they're going to hit it. If you have one that, like if there's a lot over here, then the probability of this being hit by a potassium ion is highly likely, and it will follow down the trail. Where if there aren't too many over here, hitting it is low. So that's the gradient basis. The water, even though draw it to the wall, it can only interact with maybe one, or with any of the others to draw it. It can't interact with them.
See, now you can see how a sodium ion, see if it is there. whether it can interact with oxygen ions in the selectivity
and
filter.
with all the water or with any of the oxygens attached to the wall. It can only interact with maybe one at the most, and that's not enough to counter its positive charge. Remember, oxygen is electronegative, so these are negative,
All the
but
oxygen
they
is
have to be
negatively
close to each
charged.
other for them to cancel each other's charges. And these are slightly negative. Can't remember the symbol. Slightly negative. They're not actually fully negative. They're slightly negative. Where this is a positive charge, this is a positive charge, and this is a positive. That's why you need 4 oxygens for our simplified case to balance out this fully charged positive ion. And let's pretend, let's for say some reason that we could even get water into this. We still probably could not get 4 oxygen atoms touching this. So let's just try it, see what happens. So we'll draw maybe one right there. Can't fit there, that's too tight. And maybe right there. But the
So
oxygen
this
doesn't.
is what in nutshell how a selectivity filter works and how it permits the movement of only sodium ions, potassium ions, and not sodium ions. Is it clear now? All three? Any clarifications needed? Okay. Then let me see whether what I can conclude. Okay, let me try to complete calcium channels also. Don't worry, it's uh, very little to understand about calcium channels. So this calcium channels, usually where is calcium channels located? Have you studied where? Muscles. Yes, it is mainly found in muscles, though rarely it is seen in neurons also. These calcium channels are slightly permeable to sodium because the size of sodium and size of calcium is almost similar. So it's slightly permeable to sodium also, and the concentration of calcium ions is very high outside the cell than inside the cell. It's almost about 1,000 times higher concentration outside the cell than inside the cell. So the calcium concentration is very high outside the cell, and once this calcium channels open, because of this gradient, ion gradient, higher concentration of calcium outside and lower concentration of calcium inside, once the calcium channels open, the calcium ions move from outside into inside the cell. And see what happens is that I told you there is a ligand gated potassium channel in the type of fourth type of potassium channel. In that ligand gated potassium channels, I told you it responds to calcium. So opening of these calcium channels and movement of calcium channels into it all leads to the activation of ligand gated potassium channels. So that is one thing about calcium channels. Then another thing is but what are the you told us that last class also what are the insecticides which acts on rhinodine receptors? Which are the insecticides acting on rhinodine receptor?
Diamide.
Diamide. Okay. So it binds to the receptors on the membrane, cell membrane, this rhinodine receptors in the muscles. So once this rhinodine receptor, the insecticide binds to this rhinodine receptor, this opens, the channels open, it permits the movement of calcium ions because we have very high concentration of calcium ions outside the cell. all these calcium ions move inside. And if you see a muscle cell, the endoplasmic reticulum of muscle cell is called the sarcoplasmic reticulum. You might have studied. The cytoplasm is called the sarcoplasm. The plasma membrane is called the sarcolemma. All those things you might have studied. The muscle cell. The endoplasmic reticulum of muscle cell is called a sarcoplasmic reticulum. This sarcoplasmic reticulum is another storehouse of calcium. There are a huge quantum of calcium ions in this sarcoplasmic reticulum. So once the calcium ions move from outside of the cell because of the binding of say insecticide or any ligand, the calcium ions open, the rhinodine, that is the receptors open, calcium ions move from outside into the cell, and the movement of calcium ions into the cell leads to the release of calcium from sarcoplasmic reticulum also. So what happens is that already calcium ions is out higher concentration outside the cell. The channels open, calcium ions move inside. So inside calcium ion increases because of the presence of higher concentration of calcium ions in the sarcoplasm. The endoplasmic reticulum or the sarcoplasmic reticulum also releases calcium from its store into the sarcoplasm. It is called as calcium induced release of calcium. Calcium concentration increases inside the sarcoplasm, and the increase in the level of calcium ions in the sarcoplasm leads to the release of calcium from the stored calcium in the sarcoplasmic reticulum into the sarcoplasm. So this is called as calcium induced calcium release. This is what happens in muscle cells normally. And if you see the structure of calcium channels, we have two alpha subunits, one beta, one gamma, and one delta subunit. The alpha subunit, one alpha subunit, the structure is similar to sodium and potassium, and I'll show that. See, this is one alpha subunit. You can see domain 1, domain 2, domain 3, and domain 4. This is the second alpha subunit, alpha 2. Then we have beta subunit, we have gamma, then we have delta subunit. So the alpha 1 subunit is structure is almost similar to sodium and voltage gate sodium and potassium channel. Any clarification you need with respect to calcium channels?
Sir, alpha 1, 2, 3, 4.
No, alpha 1 and 2, beta and gamma. We have only two alpha subunits, beta, gamma, and delta. Alpha 1, alpha 2.
Sir, the 4 domains for the
In the
sir, 4 domains for the like
it is alpha 1, this is alpha 2, this blue one, this one is alpha 2, this one is all this one is alpha 1. In alpha 1 we have D1, you can see the D1 domain 1, we have the domain 2, we have the domain 3, and we have the domain 4, and you can see the transmembrane segments 1, 2, 3, 4, 5. Here it's the sixth one is not seen, but you can, if you see it over here, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. So that's how this portion is enlarged over here. Understood? Yes. Okay. Fine. Hope you are following this. I don't, learn. I wish to state you that we had when I was doing this. There was a course in my PhD in IRA. We have a course called as Recent Advances in Entomology. Usually, it will be taken by 4 or 5 scientists considered as one of the most talented scientists in the division of entomology. So our retired head Dr. G.T. Gujjar used to take this ion channels. Specifically, Dr. Gujjar's topic was ion channels. So he used to tell about sodium channels, potassium channels, calcium channels, and all. At that time, I didn't recognize the importance of it. Uh, so later, when I tried to link this with toxicology, how this act with toxins, how it acts with different pesticides, then only I understood the relevance of this ion channel. And now understanding an ion channel is not that bit tough. It's pretty easy. Earlier we used to refer those Kirkwood and Gilbert book and all, but that is extremely complicated one to read. Didn't and nowadays if you want to read anything, internet is very good option, and YouTube we have several resources and classes from the most renowned scientists in that particular field. In sodium channel, I have I uploaded a video of Catterall, William Catterall. And William Catterall is the one who has characterized sodium channels, especially from prokaryotes, from a bacteria or something. So we can learn from the person who is exactly working on that particular field. So nowadays, learning is not that a difficult task. Okay, go through it. Today I will upload more, especially with respect to electron transport chain and all. You can go through it. Make a bit. Hopefully, I will try to take class every alternate day. Okay. Hopefully, we'll take a class on Wednesday. Okay. Thank you.